The cells in our body are constantly dividing through a process known as cell division, where we start off with a parent cell and we divide into daughter cells. Now, this is the process of cell division that allows for growth and development in species. And it's all thanks to a process, an overall process that we refer to as the cell cycle. Now, the cell cycle has two main categories. We have interphase and we have mitosis. Now, most of us are familiar with mitosis as being the process in which cell division will actually occur. And that is true. Mitosis is where we will actually see one cell divide into two cells. But then what is interphase? Interphase is going to be made up of three subcategories. It's made up of G1, S, and G2. And together, these three categories make up interphase, also known as the process in which the cell is going to be preparing for cell division. So interphase is when the cell is preparing for cell division, and mitosis is where cell division will actually occur. And together, interphase and mitosis make up the cell cycle. So once again, to clarify, the cell cycle is made up of interphase and mitosis. Interphase is when the cell is preparing for cell division, and mitosis is where cell division will occur. Now, let's take a look at these three subcategories in interphase to see what's happening in these sections and why they're important for the preparation of the cell division. Now, in G1, this is where we are going to see cell growth occur. And by cell growth, I mean that the cellular contents within the cell will duplicate, and that includes our organelles. So our organelles will duplicate in G1. So after our organelles have been duplicated and our cell is growing and preparing for cell division, it will now move on to the S phase. And in the S phase, that is where we will see DNA replication occur. Now, one thing we need to understand is that in all human somatic cells, we will have 46 chromosomes. So if we are going to undergo mitosis and we're going to produce two daughter cells, we need to make sure that both of those daughter cells end up with 46 chromosomes. So in order to end up with 46 chromosomes in each daughter cell, we must duplicate our DNA. So taking that into consideration, this brings up a very important concept that students often mistake. Now, when we look at DNA replication, we start off with this structure over here. Now, this structure is a chromosome, but we can also refer to it as a chromatid. But if we take a look at this structure over here, this can also be referred to as a chromosome. But this chromosome is made up of two chromatids. To review one more time, so this can be a singular chromatid. It can also be called a chromosome. But this chromosome is made up of two chromatids. And when a chromosome is made up of two chromatids, we call these two chromatids sister chromatids. So when I say that in the S phase we're undergoing DNA replication, essentially we start off with 46 chromosomes, or we can refer to them as 46 chromatids, and then we replicate the DNA and we still have 46 chromosomes, but now we're going to have sister chromatids. So overall, we're going to have 92 chromatids after the S phase where DNA is replicated. Once again, we are going to start off with 46 chromosomes or chromatids. We're going to duplicate the chromatids and we're going to end up with 92 chromatids, but we're still only going to have 46 chromosomes at this point. Now, one more thing I want to go over, since we're already looking at this diagram, is another concept that students often mistake. And that is this structure right over here. Now, at the center of a chromosome, we have specialized proteins called kinetochores. And kinetochore proteins will play a key role in the mitotic phase of the cell cycle where we see cell division occur, because it is at the kinetochore where our microtubules are going to attach. Now, this over here in blue is going to represent our kinetochore. But the region at which the kinetochores exist on a chromosome is called the centromere. So I'll write this out. So let's see. Centromere. That is the location at which the kinetochore proteins will exist on our chromatid. All right, now let's go back to look at our cell cycle. Now, once again, we started off in G1 where we saw cell growth occur, our organelles duplicated, and we moved into the S phase where DNA replication occurred. Now we're going to move into G2. Now, G2 is just the phase where we're going to see final preparations, where we see more growth happening, and the cell is essentially just making any final preps that it can before it moves into the mitotic phase. Because remember, when we're going through G1, S, G2, we're preparing for mitosis. We want to make sure that this cell has everything that it needs in order to undergo cell division. We do not want a faulty, unprepared cell going into mitosis. So after those final preparations that occur in G2, we move into mitosis. And in mitosis, we see various steps: prophase, metaphase, anaphase, telophase, and cytokinesis. And the end product of mitosis is going to be two identical daughter cells. And those daughter cells are going to be identical to the parent cell as well. Now, let's take a moment and look at some other key points within the cell cycle. And let's look at G0 for a second. So the G0 phase it's a subcategory of the cell cycle, and it essentially denotes the point in which the cell is going to be at rest. So when a cell is at rest, it is not preparing to divide. It is just simply carrying out its function. So common cells that are found in G0 are muscle cells and nerve cells. So for example, a nerve cell that is in G0 is in its resting state, so it's not preparing to divide. It's just simply carrying out its function. So the neuron will be receiving signals, sending signals. It's still carrying out its function. It's just not preparing to divide. Now, what's important to understand is for some cells, G0 is a permanent state. That means once they enter G0, they are never going to re-enter the cell cycle and undergo cell division. But for some cells, when they are in G0, they can be induced back into the cell cycle. It just depends on what kind of cell you are. For some cells, G0 is your permanent state, but for some cells can move out back into the cell cycle. That's why I have two arrows. You're entering G0, you may or may not stay there depending on what cell you are, and you may or may not come back depending on what type of cell you are. Another point we need to discuss for the cell cycle is checkpoints. Now, remember, this cell is preparing to undergo mitosis. So the cell needs to make sure that it is fully prepared and it does not have errors before entering mitosis. And therefore, we have checkpoints. Now, over here, we have our G1 checkpoint. The G1 checkpoint is the first checkpoint. Now, if the cell is going to divide and it's ready to divide, it's going to enter G1. But if the cell is not ready to divide, or it has some mistakes, or something is wrong, it will not enter G1 and it will enter G0. So this first checkpoint is very important because it essentially will determine if the cell will enter G1, will it enter G0, or it may even undergo apoptosis, which is programmed cell death. Now, once we are in G1, we've passed the G1 checkpoint. We enter S phase, and then we enter the G2 phase, and at the end of G2 phase, we have another checkpoint, and this is called the G2 checkpoint. Now, the G2 checkpoint is very important because it will essentially make sure that everything that occurred before is correct, because this is the last checkpoint before we enter mitosis, which is the cell division process. So the G2 checkpoint will make sure the DNA is replicated properly. Is it replicated sufficiently? Do we have replicated organelles? And if we are not ready, the cell will not go towards mitosis. But if we are ready, we will go towards mitosis. Now, once we are in mitosis, there is one more checkpoint, and that is in metaphase. So metaphase is a subdivision within mitosis. And essentially what happens at this checkpoint is we make sure that the chromosomes are aligned at the metaphase plate. Another way to say this is that we just make sure that the chromosomes are aligned in the middle and that they are attached to the microtubules at the kinetochore. So let's take a look one more time. So in metaphase, metaphase exists within the process of mitosis. We have that checkpoint, and this checkpoint makes sure that the chromosomes are lined up in the middle and that they are attached to microtubules at the kinetochore. Now the microtubules, once they are attached, that is what's going to cause the sister chromatids to separate to opposite sides of the cell. Now remember, this is a chromosome, and this chromosome is made up of two sister chromatids. But once these two sister chromatids separate, they will both become individual separate chromosomes. So once they separate, now we have two chromosomes when we started off with only one chromosome. All right, now that is the metaphase or M checkpoint. So we have three checkpoints. We have the G1 checkpoint, the G2 checkpoint, and we have the metaphase checkpoint. And that is essentially the key concepts that we should know for the cell cycle. To recap, the cell cycle has two main categories. We have interphase and we have mitosis. Mitosis is where we see cell division occur, and interphase is where the cell is preparing for cell division. Interphase is made up of the G1, the S, and the G2 phase. And then we also have the G0, which is cell rest. The cell is simply doing its function. It's not preparing to divide. An easy way to remember that the S phase is for DNA replication is that S stands for synthesis. And when we think of synthesis, we can think of synthesizing DNA, and we're duplicating our DNA. And then lastly, we have our three checkpoints, the G1, the G2, and the M checkpoint. And the M checkpoint makes sure that everything is aligned at that metaphase plate and the microtubules are attached to the kinetochore.